Welcome back. Glad to have you here for another deep dive. Today, we're going to be tackling something that's really essential in the world of medicine. We're going to be talking about phase 2 clinical trials. And, we're focusing on, I think a lot of you out there are really interested in understanding, like, how do researchers actually design these trials, especially when it comes to figuring out if a new drug actually works. So we'll be focusing on those crucial elements, endpoints, and statistical power.
Yeah, that's a great place to start. Phase 2 trials are where things start to get really interesting. They bridge that gap between, those initial safety tests that are done in phase 1 and then the larger confirmatory trials that happen in phase 3. So, this is where we really start to see if a drug can truly deliver on its promise.
I love that, deliver on its promise. It reminds us that there's so much hope riding on these trials. But before we get too ahead of ourselves, let's kind of break down what makes a phase 2 trial tick. One thing that always stands out to me is this concept of control groups. Why are they so important?
Yeah, control groups are absolutely essential because what they do is they allow us to isolate the true effects of a new drug. They let us compare what happens to patients who are receiving the new treatment versus those who are receiving either a placebo or the current standard of care or sometimes even no treatment at all. So it helps us rule out any other factors that might be influencing the results, besides the treatment itself.
Okay, that makes sense. But what about, the ethics of giving some patients a placebo? I mean, isn't it kind of a dilemma if we think that this new drug might actually be helpful?
Yeah, you've hit on a really, really crucial point. The use of placebos is always very, very carefully considered, especially in situations where there is an existing treatment available. And it's all about balancing the need for rigorous scientific evidence with the well-being of the patients who are participating in the trial. So, researchers and ethical review boards, they grapple with this constantly, weighing the potential benefits of the new treatment against the risks of using a placebo. And in some cases, using a placebo might be deemed unethical, and the new drug would be compared to the existing standard of care instead.
That's really insightful. I mean, it really helps me understand the complexities involved in kind of designing these trials. So we've got our treatment group and we've got our control group. How do we decide who goes where? I mean, isn't there a risk of bias if the researchers are kind of handpicking patients for each group?
Absolutely. And that's where randomization comes in. It's essentially like flipping a coin to decide which group each patient will be assigned to. And by doing this, what we do is we ensure that the groups are as similar as possible at the start of the trial. It minimizes the chances that any differences we see in the results are due to pre-existing factors rather than the treatment itself.
It levels the playing field, so to speak.
Mhm.
And what about blinding? I've heard that term before, but I'm not sure I fully grasp what it means.
Yeah, blinding is all about keeping people in the dark about who's getting which treatment. In a single blind trial, the patients themselves don't know what they're receiving. And in a double blind trial, both the patients and the researchers who are administering the treatment are unaware. And this really helps prevent subconscious bias from influencing how patients report their symptoms or even how researchers interpret the data.
So it's like removing, any chance of a placebo effect or researcher expectations influencing the outcomes. It sounds like these design elements, control groups, randomization, and blinding, they're all about creating a really, really robust foundation for the trial. But how do we actually measure whether the drug is working? That's where I think clinical endpoints come in, right?
You're absolutely right. Endpoints are the specific measurements that we use to determine whether a treatment is effective. And one of our sources describes them as the measuring sticks of success, which I think is a great way to visualize it. It's all about setting a target and then figuring out how to measure whether we're hitting the bullseye.
I love that analogy. So are we talking about things like survival rates
Mm.
or disease progression or even how patients, report they feel?
Exactly. There are different types of endpoints. Some of them are what we call hard or clinical endpoints, things like overall survival. Those are pretty clear cut. And then we have what are called surrogate endpoints, which might be things like, tumor shrinkage in an oncology trial or blood sugar control in a diabetes trial. They're not the ultimate outcome, but they can give us an early indication of whether the drug is having a biological effect. And that's especially useful in phase 2, where we're looking for those early signals of efficacy.
This is like choosing the right, yardstick depending on the disease and the treatment being studied, right? Are there any real-world examples from our sources that might help illustrate this for people?
There are, yeah, there was one study looking at a new diabetes treatment, and they used glucose AUC reduction as an endpoint. Essentially, this measures how well the drug controlled blood sugar over a specific period of time. Now, it's a surrogate endpoint because it's not directly measuring, how long people live or whether they develop complications, but it gives researchers a good indication of whether the drug is effectively targeting the underlying disease process.
That makes it much more concrete for me. It's like we're not just looking at these abstract concepts, but we're actually seeing how these endpoints play out in a real-world study.
Okay.
And this brings up another question for me. How do researchers make sure they have enough patients in a trial to actually detect a real effect if one exists? I've heard the term statistical power thrown around a lot, but I'm not quite sure I understand it.
Yeah, and that's a crucial point. And it ties into this concept of statistical significance. You might have heard of the p-value, which is often set at 0.05 in research. Basically, it means there's less than a 5% chance that the results we're seeing are due to random chance. Statistical power is the probability of actually finding that statistically significant result if a real effect exists. And the more patients we have in a trial, the greater our power to detect even small effects.
So if a trial is underpowered, it's like trying to find a needle in a haystack with a tiny magnet. We might miss a real, potentially beneficial effect of the drug just because we didn't have enough patients to see it clearly.
Exactly. And that's why sample size is such a key driver of statistical power. Of course, the magnitude of the effect we're looking for also matters. If the drug has a huge impact, we might be able to detect it with a smaller sample size. But in many cases, the effects are more subtle, and we need a larger group of patients to be confident in our findings.
This is all starting to click for me now. We talked about the core elements of good trial design, the control groups, randomization, and blinding, and how they create a solid foundation for getting those reliable results. Then we kind of dove into this fascinating world of clinical endpoints, those measuring sticks that tell us whether a drug is working. And now we're kind of starting to grasp the importance of statistical power in making sure our trials are sensitive enough to detect those real effects if they exist. But before we move on, I'm curious, what stands out to you so far? What's sparking your curiosity? Hold on to that thought, and we'll pick back up in part 2 with even more insights into the world of phase 2 clinical trials.
It's fascinating how much actually goes into designing these phase 2 trials, isn't it? We've talked about controlling for bias and selecting those right endpoints to measure success. But there's another layer here, I think, that's essential for making sure that these trials are conducted to the highest possible standards, and that's the regulatory landscape.
You're right, we can't just design these trials in a vacuum, can we? There are guidelines and regulations that researchers need to follow.
Absolutely. Organizations like the FDA here in the US and the ICH, the International Council for Harmonization globally, they provide a framework for the ethical and scientific conduct of research. Throughout all phases of drug development, so think of them as the guardians of patient safety and the gatekeepers for new drugs reaching the market.
So it's not just about good intentions, right? There's an entire system in place to make sure that these trials are done right. What kind of guidance do these organizations provide, specifically for phase 2 trials?
Well, their guidelines cover a wide range of aspects. For instance, there's the ICH E9 guideline, which focuses on statistical principles. It emphasizes the importance of having a clear scientific hypothesis, selecting appropriate endpoints, and then determining an adequate sample size to ensure sufficient statistical power.
That makes sense. I mean, we already talked about why statistical power is so crucial for detecting real effects. But it's interesting to know that this isn't just left up to chance. There are guidelines to make sure that researchers are doing this rigorously.
Oh, absolutely, and there's more. The ICH E6 guideline provides a framework for good clinical practice throughout the entire trial process. It covers everything from obtaining informed consent from patients to ensuring the accuracy and integrity of the data collected.
So it's like a comprehensive roadmap for conducting ethical and scientifically sound research.
Yeah.
Do these guidelines also address the specific challenges of different therapeutic areas? I mean, a trial for, let's say, a new heart medication probably has different considerations than one for a new cancer treatment.
You're spot on, and both the FDA and the ICH have developed a whole library of guidelines tailored to different disease areas. For example, there are specific guidelines for conducting trials in oncology, cardiovascular disease, infectious diseases, and so many more. And these guidelines really reflect the unique challenges and considerations inherent in each of these areas.
So it sounds like a very complex web of regulations, but ultimately it's all about ensuring that these clinical trials are conducted to the highest possible standards.
Right, precisely. And that's ultimately for the benefit of patients. By adhering to these rigorous standards, we increase the chances of developing safe and effective treatments that truly improve people's lives.
That brings us back to why we're doing all of this in the first place, right? It's not just about scientific curiosity, but it's about making a real difference in people's lives. We talked earlier about statistical power and how important it is to have enough patients in a trial to detect a real effect, if one exists. What are some of the challenges that researchers face in getting those calculations right, especially in phase 2?
That's a great question. And one of the biggest challenges in phase 2 is that we're often dealing with smaller sample sizes compared to those massive phase 3 trials. This means we have to be extra careful about our assumptions going in.
What kind of assumptions are we talking about here?
Well, one crucial assumption is the estimated effect size of the drug. So, how much of a difference do we think it will make compared to the control group? If we overestimate that effect size, we might end up with a trial that's underpowered, and we could miss a real effect, even if it's there.
So it's like kind of setting the bar too high and then being disappointed when the drug can't clear it.
Exactly. And on the flip side, if we underestimate the effect size, we might end up with a trial that's overpowered, meaning we enroll more patients than we really need, which wastes resources, and it could potentially expose more patients to the risks of a new treatment unnecessarily.
Sounds like a delicate balancing act.
hmm
You need to find that sweet spot where the trial is powerful enough to detect a real effect, but not so large that it becomes unmanageable and expensive.
Exactly. And it's not just about effect size. We also have to consider the variability in the data that we're collecting. And this can be influenced by things like the patient population, the disease being studied, and even the measurement methods that we're using.
Right, right.
So getting these assumptions right is really crucial for making sure that we have enough patients to see a clear signal, if the drug is truly working.
This highlights how important it is to be, I guess, a critical reader of research, right?
Hmm.
Just because a trial shows a positive result doesn't necessarily mean it's a slam dunk. We need to understand how those results were generated and whether the trial was designed in a way that gives us confidence in those findings. That's like being a detective, looking for clues to see if the evidence really stacks up.
I like that analogy. And speaking of looking closely, let's talk about some common misconceptions about phase 2 trials. A lot of people, when they hear clinical trial, they think of these huge, late stage studies. But phase 2 is a different animal altogether.
Okay, so what sets phase 2 apart?
Well, phase 2 trials are often smaller and more exploratory than those large phase 3 trials. Their primary goal is to gather that preliminary evidence of efficacy and to identify the optimal dose and dosing regimen for the drug. Think of it like, fine-tuning a recipe before you bake a giant cake.
So it's not necessarily about proving beyond a shadow of a doubt that the drug works, but it's more about getting those early signals and figuring out the best way to administer the treatment.
Exactly. Phase 2 trials are designed to provide a signal, a hint that the drug might be effective. They help us narrow down the possibilities and determine whether it's worth moving forward to those larger, more expensive phase 3
Oh
trials.
boy.
Sometimes a phase 2 trial might show that the drug isn't as effective as we initially thought, or that it has unexpected side effects, and in those cases, it might be decided to stop the development altogether, which saves time and resources.
It sounds like phase 2 is all about gathering that crucial information to inform those critical gonna-go decisions in the drug development process.
Precisely. And that's why these trials are so valuable. They help us weed out the less promising candidates and focus our efforts on the treatments that have the greatest potential to benefit patients.
We've covered a lot of ground in this part of our deep dive, haven't we? We've explored the regulatory landscape that kind of governs these trials, tackled some common misconceptions about phase 2, and delved deeper into those complexities of statistical power. But our exploration isn't over yet. Hold on to those insights, and we'll pick back up in part 3 with even more fascinating discussions about the world of phase 2 clinical trials. Welcome back to our deep dive into this fascinating world of phase 2 clinical trials. We've already covered so much ground, from those core principles of trial design to the complexities of statistical power and the crucial role of those regulatory guidelines. But I have a feeling the conversation's about to get even more interesting as we kind of explore some of those cutting edge trends that are really shaping the future of drug development.
Yeah, you're absolutely right. The field of clinical research is constantly evolving, and phase 2 trials are really right at the forefront of that evolution. We have new technologies, innovative therapeutic approaches, and a growing understanding of human biology. All of those are influencing how we design and conduct these studies.
Okay, I'm all ears. What are some of the trends that are really, shaking things up in this world of phase 2 trials?
Well, one of the most exciting trends, I think, is the rise of personalized medicine. It's really a paradigm shift from that traditional one-size-fits-all approach to medicine. We're really moving toward treatments that are tailored to an individual's unique genetic makeup, their lifestyle, and even their environmental factors. It's kind of like the difference between buying a ready-made suit and having one custom tailored just for you.
I love that analogy. So how does this personalized approach actually impact the design of phase 2 trials?
Well, it's changing everything. Instead of
Huh.
enrolling large, diverse groups of patients, we're starting to see smaller, more targeted trials that focus on specific subgroups of patients who are most likely to benefit from a particular treatment.
That makes a lot of sense. It seems more efficient and potentially more ethical too, since you're not exposing patients to treatments that are unlikely to work for them.
Exactly. And this trend is really closely linked to the increasing use of biomarkers, which are essentially measurable indicators of a biological process. And by using these biomarkers to identify those patients who are most likely to respond, we can increase the efficiency and the success rate of our trials.
So biomarkers are like clues that help us predict, I guess, which patients are most likely to benefit from a specific treatment. It's like having this, personalized roadmap for each patient's journey through the trial.
That's a great way to put it. And the use of biomarkers is particularly relevant in areas like oncology, where there's this growing emphasis on matching patients with the most effective, chemotherapy regimens or targeted therapies.
It's incredible to think about how much progress has been made in cancer treatment, over the past, couple of decades. I'm guessing personalized medicine and biomarkers are playing a huge role in those advances.
Absolutely. Immunotherapy, which harnesses the power of the immune system to fight cancer, is a prime example of personalized medicine in action. These treatments are often tailored to the specific genetic mutations that are present in a patient's tumor, and this can really increase their effectiveness and minimize those side effects.
It sounds like we're moving towards a future where treatment is less about trial and error, and it's more about this, precision targeting.
Yeah.
I'm curious how these trends are influencing the selection of clinical endpoints in phase 2 trials.
That's a great question. And with personalized medicine, we're seeing a greater focus on endpoints that are more directly relevant to the individual patient rather than just overall survival. For example, in a trial for a new cancer treatment, we might focus on progression-free survival, which measures how long a patient lives without their disease worsening. Or, we might even prioritize quality of life measures that assess how the treatment impacts a patient's daily functioning.
So it's not just about adding years to life, but adding life to years,
Yeah,
right? It's a more holistic approach to measuring success.
exactly. It reflects this growing recognition that patient-centered care is paramount. It's not just about extending life, but it's about improving the quality of that life.
Well said. And speaking of groundbreaking advancements, we can't forget about gene therapy. It seems like it has the potential to just revolutionize how we treat a wide range of diseases.
Gene therapy is incredibly exciting, and it involves modifying a patient's genes to correct a defect or to introduce a therapeutic gene. It's kind of like, rewriting the code of life itself.
That's a really powerful image. But I imagine designing these clinical trials for gene therapies, it presents some unique challenges, right?
Absolutely. One of the biggest challenges is determining the durability of the treatment.
Yeah.
Will that genetic modification persist over time, or will the therapeutic effect eventually wear off? And this is a crucial consideration when we're selecting those endpoints and designing these trials.
So it's not just about seeing that initial response, but you have to track those patients long term to make sure that the treatment has lasting benefits. That must be incredibly complex, especially for rare diseases where you might have a very limited number of patients to enroll in these trials.
You're right. It's a complex endeavor, and that complexity really just highlights the constant evolution of clinical research. Researchers need to be creative and adaptable in their approach, always seeking new ways to measure success and ensure patient well-being.
This has been an incredible journey. We've gone from those foundational elements of trial design to the cutting edge of personalized medicine and gene therapy, and it's clear that phase 2 trials play this pivotal role in bringing new treatments to those patients and pushing those boundaries of medical innovation.
I completely agree, and it's important to remember that these advancements are built on a foundation of rigorous science, ethical conduct, and a deep commitment to patient well-being.
Thank you so much for sharing your expertise with us today. You've given us, I think, a much deeper understanding of all the intricacies of these phase 2 clinical trials and the incredible effort that goes into developing those safe and effective new treatments.
It's been my pleasure. And remember, knowledge is power. The more we understand about clinical research, the more informed decisions we can make about our own health and the future of medicine.
And until our next deep dive, keep exploring the world around you with curiosity and a thirst for knowledge. There's always something new to discover.